Hi students, welcome back to the third part of nutrition in humans. Okay, so we'll be looking at the characteristics that is present in ileum itself, since we have done digestion all the way up to the small intestines. So the ileum, a recap, it is the main site of food absorption after food has been digested down to the smallest form, as you've seen by all these green color components here. Okay, so why is it possible that the ileum is able to carry out this is due to the finger-like projections, which increases the surface area to volume ratio. Okay, so each of these finger-like projections, they also contain lacteals, which is used for absorption of different types of food components. Okay, so when we move down, what we see over here before we look at the table itself, we have got the villus component of the small intestines. Okay, so let's say I do a cross section of the small intestines and I take out a certain small little hump over here and I expand it out. I will actually have this larger region. So you can see that when one of these small little humps, they are expanded, on top of each and every small little component, we have got these little villus. Okay, so these little villuses are actually made up individually of epithelial cells, okay, and also lacteals in each of them. So in other words to say, the original two little humps, each individual humps, they are made up of hundreds of tiny little cells, each comprising of a lacteal itself and epithelial cells. Okay, so on the epithelial cells, we have got these little microvilli, which will increase the surface area to volume ratio because these are the finger-like projections. Okay, so another thing that you can see about these epithelial cells is that they are only one cell thick. Okay, so because they are only one cell thick, that will increase the diffusion rate of the substances. Okay, so the villi and the microvilli itself, which are these little protrusions, they will increase the surface area to volume ratio. Okay, so even though the small intestines just look like one rope of component or one rope of organ in the body, but because of its adaptations and its structural components, when you spread out the entire thing, it is actually about 7 meters long. Okay, so 7 meters long in total will allow rapid absorption of the digested food substances. In addition to that, whenever we have got your glucose and amino acids that are being taken away through absorption from the small intestines, and that is made possible by the presence of the blood vessels in the regions. Okay, so by transporting the sugars and amino acids, that will enable a concentration gradient, which will rapidly enable diffusion to take place. Now, what about my fats itself? Now, fats, they are removed by the lacteals, or rather transported by the lacteals, which is this middle component that you see here, and they are all surrounded by blood vessels around it. Okay, so the small intestines also carries out peristalsis. So peristalsis was carried out in the esophagus, as you saw in the first video. Now the action of peristalsis in the small intestines enable the food to be brought closer to these intestinal walls, and at the intestinal walls I have got all my microvilli and villi component. Okay, now what happens after food is being digested and transported by the small intestines is that these small intestines component, the blood capillaries, will then transfer it to what we call the hepatic portal vein. Okay, hepatic because it refers to the liver, vein because it is not supplying blood directly from the heart itself. Okay, portal because it acts as a transportation. Okay, now what about your fatty acids and glycerol itself? So for fatty acids and glycerol, they will enter into the lacteals as we saw earlier on, and from the lacteals it will then enter into the lymphatic vessels, whereby it carries throughout the circulatory system itself. Okay, so coming down to the next part here, transportation and assimilation is what we call for hepatic portal vein. So firstly, there is transportation from the small intestines of your simple sugars, your monosaccharides, okay, your glucose, fructose and galactose, along with your amino acids. So these will then enter into the liver itself. So when it enters into the liver, some of it will undergo deamination, some of it will be distributed. Okay, so for simple sugars itself, the simple sugars, they are oxidized for respiration. So in the process of respiration, oxidation is the process that carries out respiration itself. What happens is that ATP is produced, okay, and ATP is the cell currency for energy. So for all the cells that require energy, they will have to produce ATP, and this is done by an organelle, the mitochondria. Okay, for this, refer back to your earlier topic 1 on cells itself, whereby we went through mitochondria. Okay, now in the liver, what happens when you eat, for example, too much carbohydrates? So you have got too much glucose, and these components, the excess glucose, will then be converted into glycogen. Okay, so recall earlier on I said the pancreas is able to produce not only enzymes but also hormones. Okay, so one of these hormones is insulin. So insulin, I'm sure some of you have heard it before, whereby diabetic patients, they require either insulin medication that they eat orally, or in more bad scenarios, these patients require injection of insulin after every meal. Okay, so what insulin does is that they prevent excessive sugar or glucose from circulating in the circulatory system, which can cause a wide range of medical conditions. So instead, what insulin does is that they will convert all these excess sugars into glycogen. Okay, so once it is converted to glycogen, it is then stored in the liver. Okay, so the brain will sense that there's excess glucose, okay, and it will stimulate the production of insulin at the islets of Langerhans, at the specific area for insulin production at the pancreas. Excess glucose is then stored into glycogen. Okay, so you can see that this is different from digestion, in the sense that digestion, you are breaking it down from a large carbohydrate into a monosaccharide. Okay, in the liver itself, excess small little monosaccharides are now converted into glycogen. Okay, and this is what we know as assimilation. Okay, assimilation can also be, for example, when I have got my amino acids. Okay, now my amino acids are going to enter cells, and in the cells, what happens? You will have your ribosomes. Okay, so the ribosomes are now going to be able to synthesize proteins from the amino acids, okay, for either for repair or growth, which is what your proteins are supposed to do. So, and this entire process is also known as assimilation. So assimilation, what you are doing is that you are getting these small substances, and these small substances will then be converted into large components to be used by the cells. Okay, now moving down from this page here, I will just leave this here. Okay, so moving down from this page, this is basically what we have went through. So just briefly, excess glucose is stored in the liver. Okay, so when is it that I will need to use these excess stored components? Okay, it can be either during starvation, or it can be during, for example, a voluntary fasting. Okay, or it can also be due to the presence of this hormone called adrenaline. Okay, so adrenaline, we will look at it again later on. So adrenaline briefly is produced by adrenal glands, and adrenal glands are located at the top of your kidneys. Okay, so adrenaline is usually produced, for example, when you are in a very nervous situation. Okay, and this causes excess pumping of the heart. You feel the blood rushing through your veins. You feel that your sense of hearing and your sense of sight has increased, etc. And all this is made possible due to adrenaline. So when the body readies itself for additional action to be taken, what happens is that you need more energy. So how do you get more energy? You get it from glucose. Okay, so your body is now going to convert all these stored glycogen into glucose, which is then utilized by the cells. Okay, so that wraps up for the component on glucose. Now what about amino acids? So excess amino acids, they are also transported to the liver via the hepatic portal vein. Okay, so all these excess amino acids, they undergo this process which is known as deamination. Okay, so deamination is a process whereby you break down the amino acids into its individual structural components. So now let's take a look at this one. This is your general structure of an amino acid. Okay, we have got 20 different amino acids. You are not required to know these 20 different amino acids. They are usually denoted by this R group to indicate that there are 20 different ones. Okay, all amino acids have the same backbone structure, in which you have got a carboxyl group at one end and you have got an amino group at the other end. Okay, so in the liver, the carboxyl group is converted into glucose and excess glucose is converted into glycogen. Now what about the amine group or the amino group? Okay, so this, because it contains nitrogen, is converted into ammonia. Now ammonia is extremely toxic to the body, okay, which is why ammonia is very rapidly converted into urea, which is then able to be removed out through the urine via the kidneys, and in that we call that the excretory system. Okay, so whereby you have got excretion that takes place, which is the removal of urine from the body itself. Okay, now important functions of the liver. Okay, so some important functions of the liver here, some of it that we have gone through. So let's go through the ones that we have gone through first. We went through your metabolism of glucose, okay, whereby excess glucose is being stored in the form of glycogen. Metabolism of fats. Okay, so how is it that the liver is responsible for fat metabolism? Is due to the production of bile, okay, and bile will emulsify fats, and the liver is an accessory organ to that. So similarly for proteins, it will undergo deamination. Okay, now this part on plasma proteins is not in the syllabus. It's FYI in the event that your school goes through it. Okay, some of these proteins, the important one that you will see in your syllabus is fibrinogen, which is important in clotting, and you will see that under the circulatory system topic itself. Okay, now the liver is also the site of detoxification. So whatever perfume smells, your Panadol that you take, or some food that does not agree with you, etc., all these will go to the liver for detoxification. Okay, one classic example is alcohol. Alcohol does not agree with most people. So what happens is this enzyme called alcohol dehydrogenase will break it down into carbon dioxide and water, which can be removed safely from the body. Okay, so some individuals do not contain this enzyme at all, or they have got low quantities of this enzyme, and more often than not, they find that they are extremely allergic to alcohol itself. Okay, the liver acts also as a storage site, contains all these vitamins. Okay, folate is a type of vitamin B, okay, under the class of vitamin B itself. It also carries the red blood cells after destruction. Okay, so recall your red blood cells have only about 120 days to live. Okay, 120 days due to the lack of the nucleus. Okay, so when it is broken down in the spleen, spleen, okay, what happens is that the hemoglobin component is sent to the liver, and the liver will then store the iron. Okay, which is why in some cultures, most people, they are recommended to eat the liver itself to increase the amount of red blood cells. Okay, heat production. The liver carries out lots of chemical reactions. These are just a few. So you can see that the liver is put through a huge amount of use every single moment. Okay, so due to that, you have got heat production, and the heat is then transported to other parts of the body. Okay, now that's the end of this lecture. The next part of the lecture, we will look at the other components of this chapter, nutrition in humans, such as the effects of alcohol. Thank you for watching.